This is a series of videos on integration of metabolism. At the end of looking at carbohydrates, proteins and fatty acid metabolism in detail, we should understand that metabolism, integration of metabolism is dependent on many organs. When we speak about organs, organs are made up of tissues. There are organs that are made up of different types of tissues. For example, you look at kidney. Kidney will have medullary region and the cortex region. And tissues are made up of cells. So different types of cells can be present in a tissue. But what is interesting is all these cells will behave for a common purpose. That is something amazing about a tissue or an organ. All cells in our body are genetically, it is the same. However, they are very different in their response to hormones, their expression of proteins or enzymes, and how they will behave in response to a particular fuel molecule. In order to look very deeply into this, we are going to look at every organ that is associated with the integration of metabolism. We will begin this discussion starting with the liver. So, as we know, liver plays a very key role in integrating metabolism. When you look at the structure of a liver, it is somewhere around 1 to 1.5 kilograms. That is the size of a liver. And it is placed just beneath the diaphragm. And liver has varieties of cells present. 80% of liver is made up of cells called hepatocytes. So hepatocytes with a very typical cuboidal shape. And the remaining 20% is made up of varieties of cells. Most important them is they are very small. They are the Kupffer cells. And these cells are nothing but the macrophages. And they occupy very small volume in liver in comparison to the hepatocytes. Hepatocytes are mostly involved in metabolic reactions. Let us look very deeply into arrangement of the cells as well as the kind of blood supply to the liver cells. One of the most important is the hepatic portal vein. Now, hepatic portal vein, which is only about 7 to 8 centimeter long, that will connect in the small intestine. After all the nutrients are absorbed, it is directly being transferred to liver. So therefore, all the protein which is converted to amino acid and carbohydrate, the monosaccharides, will be directly delivered to liver. Whereas the fatty acids will be transferred to lymphatics and it will be circulated throughout the body. What exactly happens when a sugar, a carbohydrate reaches liver? Primarily, we need to look at the absorption of this carbohydrate. Remember, liver has GLUT2 protein which helps in the which is supposed to help liver to take up glucose molecules. But GLUT2 is non-responsive to the glucose level. Saturation level of GLUT2 is very high. So therefore, at any given point of time, GLUT2 remains unsaturated and from a high concentration, glucose can be taken up by liver. Look at the kind of mechanism liver has adopted in order to absorb maximum glucose. Once the glucose is absorbed, liver has an important enzyme to convert glucose to glucose 6-phosphate. Now, this conversion is carried out by glucokinase enzyme. This is an enzyme present in liver. It is also called hexokinase number 4. Hexokinase number 4. Now, this enzyme has a Km value which is greater than 10. So, greater than 10 millimolar. It means this enzyme, it is difficult to saturate this enzyme. So, all the glucose that enters into the hepatocytes will immediately be converted to glucose 6-phosphate. Remember, this is, we are talking in comparison to a hexokinase which has a Km value very low, 0.01 to 0.05 millimolar. So, it will easily get saturated, whereas this gives a protective function to our body, especially in the post-absorptive period. So, once glucose 6-phosphate is made in the liver, there are 3 different fates for this. One possible fate of glucose 6-phosphate is to convert this into glycogen. So, about 75 to 80 percentage of the glucose which is being absorbed is converted to glycogen, and it will be stored. Very little is stored in liver, most of it is stored in muscle. Another fate for glucose 6-phosphate is it will undergo oxidation and pyruvate will be formed, and this pyruvate will enter into TCA cycle by forming acetyl-CoA, and it will produce energy. So this energy can be used by liver, but liver depends on the energy that is coming from amino acid degradation or fatty acid degradation for routine activity. So only about 25 or 20 to 25% of glucose is getting into this form, and the third fate of glucose 6-phosphate is convert glucose 6-phosphate through the pentose phosphate pathway. So it will form ribulose 5-phosphate, and it will also produce NADPH, which is needed for several biosynthetic reactions. So look at the functions of liver, especially with regard to the carbohydrates. What happens to the amino acid which are absorbed by liver? Most of the amino acids are used for protein synthesis. We should understand that amino acid, liver produces about 30 grams of urea per day. This is an important function of liver. So, the extra amino acids will be deaminated and it will be converted to urea and it will be excreted. Otherwise, liver use all the amino acid for protein production. What about fatty acids that are being absorbed? In the fatty acids that are absorbed, generally it is used for synthesizing triacylglycerol and used for storage purpose. Liver is an important site for the production of fatty acids. So, liver also oxidation of fatty acid occurs in liver mitochondria. Now, when synthesis of fatty acid occurs, we know that from previous videos, the first product which is formed is malonyl-CoA. So malonyl-CoA will be formed from acetyl-CoA by using an enzyme known as acetyl-CoA carboxylase. Now this malonyl-CoA has an inhibitory effect on we represent a minus sign inhibitory effect on an enzyme known as CAT1, carnitine acyltransferase 1. This is the enzyme responsible for transferring of fatty acyl-CoA into the mitochondria. So therefore, entry of fatty acid to mitochondria is regulated by CAT1. So when synthesis has to be promoted, malonyl-CoA will inhibit this CAT1 and fatty acids will not be transported to mitochondria for oxidation. So what happens? If the fatty acids are used for now synthesis of triacylglycerol, they are packed and sent for storage purpose to adipose tissues.